Okay, can somebody confirm you can hear me?
I can hear you.
Thank you. If somebody wasn't in the lecture, you didn't know that I told you guys that I'm going to try and start this lab at 6:32. And that's because we ran a little bit over in the lecture and I kind of needed 10 minutes to do something. So anyways, are there any questions before we begin? Let me state that I do have one of the labs graded. Let me check on that and I'll tell you which one. I don't remember. I think it might be lab 3, but let me stop sharing my screen and then I'll go into the grade book and I'll be able to tell you that. Yes, lab 3 is graded. So if you did not get 100%, you may resubmit by this coming Saturday. Oh, good. The lecture just finished its file, the Zoom file, so my computer will now work better. So you can turn in the assignment for lab 3 until February 3rd. That's this Saturday at 11:59 p.m. for a second grading. I do not have lab 4 graded, and because of my Thursday class, I'm not going to get it graded anytime soon. And so probably lab 4 will not be graded until later this week. And so I will not say that the second grading has to be this Saturday. It'll probably be a week from Saturday. I'm going to make that change right now. And that's because I know I'm not going to be getting this graded anytime soon. So that one, hopefully, it needs to be turned in by February 10th, but I haven't graded it yet. So this is just still a hope. But I'd be really surprised if I got that far behind. Um, just a lot to do, and I'll try and get that graded. When can I get it graded? Maybe I can get it graded on Wednesday. All right, any question about any of that? If not, let's go ahead and get out of there. Get out of my grade book. So I can share my screen. All right, let's share my screen again. All right, if there's no questions, today is January 29th. And today we're going to be discussing Lab 5, streaking for isolation and culture media. Now you should read the unknown project instructions file on Canvas for next week. Let me show you where that is. I think it's week 5 lab. Come on. There we go. So the unknown instructions file, this will be your instructions for your unknown project. Next week, not this week, but next week, I will be talking about the unknown project and you guys will shortly be starting it sometime next week. Any question about any of that? I will mention right now in the schedule, there is nothing scheduled for Wednesday of this week. So I will be dropping in the lab for the first 15 minutes to answer any questions. And then after that, I will log out. So if you have questions in the lab or any other questions in the class, I will be there during the lab time, but only for the first 15 minutes on Wednesday. Any question about any of that? On Wednesday, instead of having the lab, you can read the unknown project instructions. And then next week we will discuss and then start the unknown project. All right, any questions? If not, let's begin lab 5. I'll blow that up a little. Lab 5, cultivation of bacteria, culture media, inoculation, and isolation. As is always, there will be some reading out of your textbook that you should do. Read about culture media and read about obtaining pure cultures. There are some video clips in today's lab. There's no virtual lab. The learning objectives for this lab are: 1, understand the importance of proper specimen collection and transport to the microbiology lab. 2, explain the 5 principal steps used by microbiologists to process specimens. 3, differentiate between the various types of different culture media. 4, explain the purpose of streak plate culture technique, also called streaking for colony isolation. And 5, define the following terms: selective media, differential media, and reducing media. Any questions about any of that? All right. The ability to isolate pathogens from a clinical specimen submitted for culture is only as good as the collection technique and the timely transport of the specimen to the lab. Some microbes are sensitive to desiccation, meaning they'll dry out, or exposure to oxygen, such as an anaerobe, and require special transport media to preserve the specimen. Once a specimen is received in the lab for biological culture, microbiologists employ the 5 principal I steps to process it. First, there's inoculation. Second, there's isolation. 3, incubation. 4, inspection. And 5, identification. Inoculation is placing the specimen on an artificial culture medium to initiate its growth. Isolation is spatially separating microbial growth into distinct colonies. Incubation is placing the culture medium in environmental conditions to facilitate their growth, meaning the growth of the microbes. 4, inspection. You then examine the culture for the presence of microbial growth. And 5, identification. Perform specific tests to ID any pathogenic microbe that has been isolated. These are the 5 steps, and we're going to be discussing the different types of culture media used in these 5 steps. You are already familiar with one media, general purpose media, such as nutrient agar or NA, or NA, nutrient agar with yeast extract, and TSA, tryptic soy agar. The general purpose media for growing many types of organisms, but they do not grow fastidious microbes. To promote the growth of fastidious microbes, you have to add certain additives to the media. So a fastidious microbe is one that has a very specific nutritional requirement. And if that nutrition is not added, the microbe will not grow. And by the way, I just recently read that recently, within the last couple of years, they've finally been able to grow syphilis in tissue culture. They've been trying to grow this organism in tissue culture for over 100 years, and they could never figure out what they needed to add to get syphilis to grow in tissue culture. Well, recently, they've figured out what to add, and now we can now grow syphilis in the lab on an artificial culture media. So that's just something I thought I would mention. And syphilis is obviously a fastidious organism, and they've been trying to get it to grow for 100 years and never succeeded until recently. So to get a fastidious microbe to grow, you need to use an enriched general purpose media. It's a general purpose media that has nutrients added, and it's called an enriched media. For example, we can add 5% sheep's blood, red blood cells, to the media to help the organism grow. And one fastidious organism that will not grow on a general purpose media, but will grow with blood agar, meaning an enriched general purpose media and containing blood, is Streptococcus pyogenes, the bacteria that causes strep throat. Any question about any of that? All right, so besides general purpose media and enriched general purpose media, there's also selective media. These media contain chemicals that inhibit or halt the growth of some organisms. However, other organisms can grow in them, and that's why they're selective. Keep in mind two things when using selective media. First, a selective media isn't necessarily perfectly selective, meaning that other organisms can grow, especially if given enough time. Second, even those organisms that are supposed to grow in the selective media may grow smaller colonies and may grow slower than they would on a general purpose media. And then besides selective media, we have differential media. These media contain chemicals that produce visible differences among different groups of organisms when the organisms are grown on that media. These differences are unrelated to how well the organism will grow on the media. It just allows you to have two different cells appear differently on the differential media. And usually it's a color difference, but it could be another difference as well. Differential culture media allows dissimilar bacteria to be visually distinguished from each other. Any question about any of that? And lastly, there is reducing media. These media promote the growth of anaerobic bacteria because low or no oxygen conditions are required to grow these organisms. And lastly, you should note that a culture media can be classified into more than one category. For example, McConkey's agar is both a selective media and a differential media. Here we're looking at McConkey's agar. It is selective in that it allows gram-negative organisms to grow while inhibiting gram-positive cells. So if the organism grows, it's gram-negative. So both these organisms are gram-negative because this is a McConkey's plate. McConkey's is also differential because if the organism grows, you can distinguish between a lactose fermenter. A lactose fermenter will turn pink, the growth on the McConkey's plate, and a non-lactose fermenter, one that does not ferment the sugar lactose, will be not pink when grown on McConkey's agar. Any question about that? Another media, which is both two types of media, is blood agar, which is both an enriched general purpose media. So you can grow some fastidious organisms on blood agar that would not grow on a general purpose media, such as Streptococcus pyogenes. And it is also a differential media because with it, you can differentiate the hemolytic patterns of the species growing on blood agar. The hemolytic pattern is the hemolysis of the red blood cells by the bacteria. You can have 3 different hemolytic patterns in cells, bacteria. You can have gamma hemolysis, where the bacteria is shown in white. Let me blow this up. It's shown in white, the white gamma here. And you can notice the red blood cells are red all the way up to the growth. And this is gamma hemolysis, and that is no hemolytic activity. There's also alpha hemolysis. The off-white is the alpha bacteria. And as you can see, the red blood cells are red up until a ring around the growth. There's a darkening or a greenish color around the bacteria. That is from alpha hemolysis, the partial lysis of the red blood cells by the bacteria. And like I said, you can see the dark ring around the A, or the kind of greenish color. It's most obvious within the alpha right there, that's all dark there. There's also beta hemolysis, and that is the complete lysis of the red blood cells. This crystal, the clear region here, is from beta hemolysis. The B of the white bacteria is the or the beta, I guess I should say, not a B, is the smear of the bacteria. And this bacteria has beta hemolytic activity, completely lysing the red blood cells around the bacteria, which makes the blood agar appear clear right there. And inside the B too. You can actually look through the plate where there's beta hemolysis. So you can hold that up to the light and see the light bulb through the beta hemolysis. You'll note that there is a ring of alpha hemolysis around the beta hemolysis, and that's normal. All right, so that's how blood agar is both an enriched general purpose media and a differential media. All right, this table is showing you the different types of media. There's the general purpose media, including TSA, NA, and NA. And then there's the differential and enriched media, such as blood agar, BAP, blood agar, which is tryptic soy agar with 5% sheep red blood cell added. And that can allow you to grow the Streptococcus pyogenes. It's also differential in that you can differentiate between the hemolytic activity of the bacteria. And then there's just a general purpose media, just an enriched general purpose media. And CAP is an example of chocolate agar. It is not differential, but it is enriched. And you can grow a fastidious organism like Haemophilus species. Streptococcus pyogenes will grow on CAP. And then there's selective and differential media. We've got three of them: McConkey's, MAC, EMB or eosine methylene blue, Hectone agar or HE. They're both selective in that they select for gram-negative bacilli. If it's not gram-negative, it generally does not grow on these three media, but if it's gram-negative, it should grow. It is also differential. All three of them are differential on the basis of lactose fermentation. They will have a different color if they ferment lactose than if they do not ferment lactose. And I'll show you that in a little bit. And then there's PEA, which is phenyl ethyl alcohol. This is a selective media. It selects to isolate gram-positive bacteria. So if the bacteria grow on a PEA plate, it's likely to be gram-positive because this media selects against gram-negative cells. And then there's selective and differential media. Mannitol salt agar is a selective and differential media. It's selective in that it selects for organisms that tolerate high salt. Mannitol salt agar has 7% sodium chloride in it, and that will select against many organisms that cannot tolerate 7% sodium chloride. So it selects for organisms that tolerate a high amount of salt, and then it's differential on its ability to ferment the sugar mannitol. If it ferments the sugar mannitol, it will turn the media yellow. If it does not ferment the sugar mannitol, it will not have a yellow color to the media, the plate. Any question about these different types of media? So first you have inoculation. That's just putting the bacteria on the culture media to get it to grow. You then have isolation. We really need to work with a peer culture, because if you are taking a culture from a patient, trying to figure out what the pathogen is, which might be causing the patient to be ill, if you don't have a peer culture, you won't be able to find the pathogen from all the other microorganisms coming from the patient. And let's say we're testing for, I don't know, hemolytic activity. You have to have a pure culture in order to do that, because if you have a pathogen and there are a bunch of other bacteria from different species, in the culture. If you have beta hemolysis, for example, you won't know which species is causing the beta hemolysis. Although I guess almost anything that causes beta hemolysis would be a human pathogen, because that's the bacteria that lyses red blood cells. Anyways, Louis Pasteur developed the concept of a pure culture, and he had a way to get a pure culture, and that was he would dilute the culture in liquid media and then replica plate the dilutions. And if he had one tube out of 10 tubes that had growth and 9 tubes out of 10 tubes did not have growth, he correctly concluded that the one tube out of the 10 with growth was a pure culture. It was a difficult way to get a pure culture. Robert Koch developed a much easier way to get a pure culture. And what he did was he streaked the bacteria on a solid plate, getting isolated colonies. And if you streak it out 2 times on 2 separate plates, meaning you pass the cells through 2 colony formation events, you will most likely have a pure culture, greater than 95% chance. Any question about any of that? All right, let's talk a little bit about streaking cultures out on a solid plate to get isolated colonies. What you do is you streak out the cells on a dish where you have quadrants or sectors. And in the first quadrant, you have a very high in density of cells. So high you have no colonies. And then after streaking out sector 1, you then sterilize your loop by putting it in the bacterial incinerator and then streaking out sector 2. That will pick up cells from sector 1 to streak out sector 2. And so these cells will be more dilute than sector 1. Still, though, that's not dilute enough because the density of the cells is so high. But you can then take your inoculating loop and put it in the bacterial incinerator to sterilize it, and then streak out sector 3. Here we're starting to get cells which might be starting to form colonies, but not quite. And then you take your loop and you put it in the bacterial incinerator to sterilize it, and then streak out sector 4. And this one is actually done correctly, so let me talk about it. What you want to do to streak out each of the sectors is take your sterile loop, put it in the previous sector to pick up cells on the loop, but you don't want to put it in where the cells are their highest density, and that would be early in sector 3. You only want to put it in, this should actually go only to here. You only want to go where the cells will be diluted in sector 3. And then streak out sector 4, coming into sector 3 at least 3 times to pick up cells on the loop. And what have we done? 1, 2, 3, 4, 5. They came in about 5 times. You wouldn't want to do it more than 5 times because you don't want to pick up a ton of cells from sector 3. You want the sector 4 to be more dilute than sector 3, and then streak it out, and then hopefully someplace on the streak, either sector 1, sector 2, sector 3, or sector 4, you'll be streaking out the cells where the cells will be plated so that a single cell will be plated on the agar to form a petri dish, not a petri dish, a colony. And in this case, that happened in sector 4, where we have isolated colonies growing up. But if the bacteria were to be less dense than what would have been, what we started with, then it wouldn't have been sector 4, which would have isolated colonies. It could have been sector 3. And if it was even less dense, the colony we started with, it could have been sector 2. And if it was very dilute cells that we got it from in the very beginning, you could even get your isolated colonies in your first sector. Any question about any of that? All right. Now, if the colonies were to be formed from one cell, that would be a pure culture. However, about 30% of the colonies will be formed from 2 or more cells. And that means 30% of these colonies are not pure. And that's too high for microbiology. So what we do is we take from one isolated colony, we will streak it out on a second plate to get isolated colonies on the second plate. And now the cells have gone through 2 colony formation events, once on the first plate and once on the second plate. If you take the cells from an isolated colony on the second plate, the chance of that being a pure culture will be higher than 95%. And so to get a pure culture, all you do is you streak for colony isolation on 2 times on the first plate, and then take from a single colony and plate it on the second plate and get isolated colonies. Any question about any of that? All right, then incubation, that's to put the media containing the cells in an environment to get them to grow. Usually, we put them in an incubator, which at the minimum can increase the temperature of the cells to grow. And we usually have our incubator set at 30 degrees or 35 degrees Celsius. And the incubator also adds humidity, which will help prevent the plates from drying out. The humidity is, we usually just have a dish in there and you add water to it. And because the incubator is heated to 30 degrees or 35 degrees, it will add humidity to the air. For the simple incubators, which is all we have in the microbiology lab in Clark College, that's all you do. You use it for warming the cells and humidifying the cells, as well as the incubator is used for a certain amount of time to grow the cells in. However, a more complex incubator shown here can control the amount of oxygen the cells will be exposed to, the amount of CO2 the cells will be exposed to, as well as the temperature, the humidity, and the time. Any question about any of that? All right. After the colonies grow up for a given amount of time, and that will be maybe overnight or after 2 days of growth, you look at visible colony morphological variations between different species of bacteria. You can look at the colonies and notice different sizes, shape, different margins, different elevation, which can help you identify the species. You cannot identify from the species from this, but you can start separating out different species. You then identify the species by running different laboratory tests. And we'll talk about different tests you can run in this lab and in much later labs. Any question about any of that? All right, know the terms of this lab. You can skip the references. In this lab, we have a video for inoculating agar plates using the streak plate culture technique. For this video, you only need to watch it until 4 minutes and 15 seconds. In this video, they only streak out 3 sectors on the plate, and that's fine. It doesn't matter if you streak 3 sectors or 4. The point is just to get isolated colonies. And then we have a little video on beta hemolysis and alpha hemolysis and gamma hemolysis, where we have bacteria being grown on blood agar plates to tell us the hemolytic activity of the bacteria. We then have a little video on McConkey's agar. On this one, you only need to watch it for 3 minutes and 50 seconds into the video. And then a little video on mannitol salt agar plates. So watch all of those videos. Unfortunately, we do not have a virtual lab for you making an actual streak plate. And that's because that virtual lab requires Adobe Flash Player to run, and so we had to pull it out. Any questions about any of this? If not, let's go to the worksheet and talk about the questions to answer for this lab. So the worksheet for lab 5, describe a natural circumstance where staphylococcal species of bacteria might benefit from salt tolerance. A natural circumstance is someplace out in the wild where Staphylococcus can grow and it is tolerating salt. Okay. 2, how might hemolysis benefit a hemolytic organism? So if you have a bacteria which can perform hemolysis, what benefit does the bacteria get from hemolyzing the red blood cells? 3, imagine you have a culture containing a single bacteria species. This culture may be one of several possible species. Describe a scenario where you use mannitol salt agar, blood agar, and EMB plates to identify the unknown bacterial species. That is, how would you use these plates to help you identify the species? And you can get 2 properties from each of these media. So for this question, you need to discuss 2 properties for each media for full credit. If you do not discuss 2 properties for each media, you will not get full credit. Four, what is the main goal for streaking for isolation? That's streaking for colony isolation. 5, what happens to the growth on the plate if you forget to sterilize the loop in between sectors when streaking for isolation? To answer this question, you need to be specific, discussing each sector. let me go to the module. So we're doing a streak here. And the question is, what happens if you have on your loop, you streak out sector 1, but then you do not sterilize the loop in between sectors 1 and 2, as well as in between 2 and 3, and in between 3 and 4. What's going to happen? So you're not sterilizing the loop in between sectors. And I will mention that you are sterilizing the loop before you begin sector 1. It doesn't say that, but you can read into it that that's what it means. So what's the growth going to look like? And you need to discuss the growth on each sector. All right, 6. What happens to the growth on the plate if you transfer from stock culture to inoculate each sectors? Be specific, discussing each sector. So here, let's come back to this one here. Instead of streaking out from stock culture for getting sector 1, and then doing sector 2, 3, and 4, you're going back to the stock culture to streak out sector 2, and then back to the stock culture to get cells to streak out sector 3, and then back to the stock culture to streak out sector 4. Tell me what the plate will look like for each sector. So you need to describe it once again, the growth on the plate for each sector. Oops, wrong one. Any question about number 5? 4, 5, and 6, sorry, 5 and 6. So that should have been anything about question number 6. Are going to be difficult. Students often get these wrong at the start. So you need to think about those. 7, suppose you successfully streak a plate and obtain isolated colonies of 2 different bacterial species. So that is, you started from a mixed culture. How can you use this plate of the isolated colonies to create a new pure culture of only 1 bacteria? Be specific in your answer. 8. In the table below are a variety of agar plate media available to microbiologists to cultivate bacteria. Denote whether each medium is non-selective, a general growth media, is selective, or a differential by placing an X in the appropriate column or columns. You may use the table of media in the lab module to answer all of these questions. So for nutrient agar, if you think it's selective, put an X there. For tryptic soy agar, if you think it's both non-selective and differential, put an X here and an X there. For tryptic soy agar with sheep's blood, that's blood agar or BAP, if you think it's non-selective, put an X there. For chocolate agar or CAP, do the same, all right? So for each of these different media, put an X where you think it should be. There can be more than 1 X for each media, but there is none which are all 3, you should have an X. Any question about any of that? All right, for number 9, it's asking you which agar plate was inoculated using a streak plate technique. Was it plate A, plate B, or plate C? All right, that's it for the lab. This will be one of your last fairly easy labs. The later labs will tend to have case studies, and those take more time and are also more difficult. But this one is difficult for questions 5 and 6. All right, go ahead and get started on the lab. I will be here until 8 o'clock to help you with any questions you have. But once the last student logs out, I will log out as well. Any questions? All right, get started on the lab. And let me stop the recording.